Hello, everyone. Welcome back to our live view. I hope that you managed to catch some cool posters at the poster session. I managed to get a couple under my belt. And welcome back for the next segment of our program, which is the presentation of the 2020 Sammy Kuo Awards for best papers from our graduate students and our postdocs over the past 12 months. And to present the awards is Professor Corinna Darian-Smith, who is in the Department of Comparative Medicine and who has been chairing the Kuo selection committees for the past 6 years. And I am so appreciative to Corinna for taking on that pleasant responsibility. Corinna's research focuses on the neural control of hand movements and recovery of these pathways after injury. And without further ado, I will turn the floor right over to my partner here, Corinna.
Thank you, Bill. So I just want to thank Bill. It's been a fabulous meeting so far, and I will be getting underway here just in a second. So I just want to start this presentation, as I always do, by giving a little bit of background about the award. Sammy Kuo was born in Taipei in Taiwan in 1984 to parents Ya Shi and Li-Ling Kuo. And in 1996, when Sammy was 12 years old, he was diagnosed with subacute sclerosing panencephalitis, which is a rare inflammatory disease arising from measles. His family cared for him at home until he passed away in 2006 at the age of 22. After Sammy's death, his cousins Peggy and Amy Chen, who were both Stanford graduates, and I believe they're both in the audience today, so welcome very much to the presentation. They contacted the Neurosciences Institute at the time and led their family to establish the Sammy Kuo Memorial Scholarship Fund to support basic research in neuroscience. This opportunity enabled the family to memorialize a boy they loved by helping to recognize crucial neuroscience research that might one day help cure other children with similar conditions. Though born of heartbreak, this award is anything but heartbreaking. It's very much a celebration, and it highlights the importance of doing basic neuroscience research that recognizes exceptional papers in early career scientists. It's, as I say, it's been going for 13 years, and the standards of the papers that are submitted are absolutely extraordinary, and I get the great pleasure of reading many of them. And if you go to the website for the Sammy Kuo, you'll be able to see all the previous winners that are listed here. It's quite star-studded. So before I get going, I'd also like to thank the selection committees for all their hard work. As I've said, I just get the lovely job of looking at the highest ranking of the submissions. They are all of excellent standard. And I also want to give credit to those working behind the scenes to make this all happen, the administrative team. I also want to apologize before I begin in case I mispronounce anybody's names. I'm not known for my excellent pronunciations. So we start this year with the Graduate Students Awards, and an honorable mention goes to Cosmos Yuki from Tom Sudoff's lab for his paper in Neuron. Cosmos used genetic manipulations of an interacting pair of transsynaptic organized molecules to probe the function of a particular circuit involved in the formation of olfactory memories. Importantly, the paper provides a new perspective on the role of centrifugal projections from the anterior olfactory nucleus to the olfactory bulb by defining the molecular basis for a synaptic connection that is key for establishing, but not maintaining or recalling, a social form of olfactory memory. The second honorable mention in the grad component of the award goes to Thamasip Khan from Sergio Pashka's lab for his paper, which came out in Nature Medicine just last week. Thamasip co-led this extraordinary effort with a postdoc, Omer Riva, who was also nominated for the postdoctoral award. This was a mechanistic study of human cortical neurons derived from patients carrying the most common human genetic microdeletion associated with psychiatric disease. Cortical organoids were derived from stem cells reprogrammed from 15 subjects with the deletion syndrome and 15 subjects who were controls. Over 400 neurons were patched and over 5,000 individual cells were imaged to demonstrate a robust phenotype across patients' cell lines and experiments. And in a clever series of experiments, the study provided novel insights into neurobiological pathways underlying the development of mental illness with this deletion syndrome. The study represents one of the largest efforts to date aimed at modeling human brain disease with stem cells. And these findings support the value of a major mutational model for elucidating the biology of complex neuropsychiatric disorders and opens a new direction for uncovering therapeutics. Our third place prize, a grad prize finalist this year, is Colin Rhodes from E.J. Chichelnitsky's lab for her paper in Neuron. In this paper, Colleen discovered striking and unusual light response properties in a newly characterized type of retinal ganglion cell in the primate retina, the smooth monostratified cells. She did a metadata analysis on 15 years of primate retinal recordings in the lab to identify a novel cell type, then focused new experiments on these cells. She found they had receptor field characteristics, hotspots of sensitivity that looked very different to textbook models of ganglion cell receptor fields. She found that the hotspots likely represent distinct portions of the dendritic branches of these cells, which act as distinct computational units that may be related to the spatial integration of inputs onto the dendrites. This work breaks new ground for our understanding of retinal processing, and her data analysis approach, which she used to identify the cells, opens the way for better understanding the diverse cell types in the primate retina. Zhang Wu from the Hong Lab is this year's second place finalist for the grad component of the Semicolo Award for his paper in PNAS. Optogenetics, as we know, has been transformative in neuroscience, but has certain limitations. There's limited tissue penetration of visible light into the deep brain structures, and it requires invasive surgery and intracranial implantations. Zhang contributed to the original design and development of what is now called sono-optogenetics, which uses mechanoluminescent nanoparticles injected intravenously. After injection in a mouse model, these nanoparticles can be charged repeatedly by 400 nanometer light while passing through the superficial blood vessels and then gated by focused ultrasound to release the stored energy locally. Zhang was able to repeatedly evoke hind limb motion in the mouse expressing channel rhodopsin 2 in motor neurons of the brain without any surgery or implantation of optical fiber or LED devices, which is really quite extraordinary. The sono-optogenetics method opens up great opportunities for neuroscience, for example, for rapid screening of different target regions in the brain for optogenetic modulation, or for any application where a light source is needed deep in the body, such as the spinal cord, peripheral nervous system, and so on. Since its publication, this technology has already received great interest both inside and outside of the Stanford neuroscience community. And this year's grad student paper of the year goes to joint co-first authors Zhefu Li and Shou Han from the Lu and Ting labs for their paper in Cell. This interdisciplinary study used in situ cell surface proteomic profiling of developing and mature olfactory projection neurons to show how the neuronal surface landscape develops over time. They designed a series of experiments in intact dissected fly brains to capture cell surface proteomes of projection neurons for mass spectrometry analysis. And they apparently led dissection parties to dissect these brains, about 9,000 fly brains. So those were some gatherings. From this analysis, they identified about 700 cell surface proteins in the pupil and adult proteomes. They found that most synaptic transmission proteins were enriched in the adult sample, whereas the pupil cell surface proteome contained many known wiring molecules. Importantly, the pupil enriched list contained proteins with no previously known function in brain wiring. And they tested the top 20 of these unknown proteins to disrupt their function and found that all 20 of them gave wiring defects, which is fairly extraordinary. This work suggests that our knowledge of wiring molecules from flies to mammals is far from complete, and it reveals many new neural wiring molecules belonging to evolutionarily conserved but previously unexpected molecular families, and opens many new avenues of research. So we now turn to the postdoctoral awardees, and an honourable mention this year goes to Omer Riva from the Pashka Lab. And for those of you with intact short-term memories, we've actually just shown this paper previously. Omer was co-lead on the same as Themisap Khan, who received the honourable mention in the grad category. As I mentioned with Themisap, this study represents one of the largest efforts to date aimed at modelling human brain disease, and in this case, psychiatric disease with stem cells. The work also shows what can be achieved with true interdisciplinary collaboration. In this case, the Themisap brought stem cell expertise, and Omer brought electrophysiology and disease pathogenesis expertise to the table. And together they uncovered the molecular and cellular phenotypes associated with the genetic form of neuropsychiatric disease. Third place this year was a tie between Sergei Stravinsky and Eleanor Cole, and we'll start with Sergei. Sergei's from the Henderson Lab, and he was first author on this beautiful study published in eLife, which demonstrated that the so-called hand knob area of the human motor cortex, which is long associated with upper limb movements, also contains representation for speech articulator movements. Using electrodes implanted chronically in the brains of research participants with paralysis, Sergei identified the neural dynamics present in motor cortex during speech and used these insights to decode a selection of spoken syllables and words with an accuracy of about 85%. This is a major advance in understanding both the organization of human motor cortex and the mechanisms by which speech is generated, and it suggests new directions for restoring function to people who struggle with communication difficulties. Eleanor Cole from the Williams Lab ties for third place this year for her study published in the American Journal of Psychiatry. It introduces a novel and effective non-invasive brain stimulation treatment for medication-resistant depression. Called SAINT, it uses functional connectivity magnetic resonance imaging guided targeting, which is able to select the optimal target region of the brain for each individual patient. She and colleagues were able to show that in just 5 days, SAINT induced remission in 90% of participants, which is almost double what is currently reported with standard treatments for treatment-resistant depression. This technique could revolutionize the treatment of depression. Importantly, it induces remission more rapidly than existing treatments, and it provides a treatment without the systemic side effects associated with many existing options. The short duration of the treatment means that SAINT could also have an application in emergency rooms or during acute hospitalizations to rapidly ensure the safety of suicidal patients. This year's second place goes to Frank Willett from the Neuroprosthetics Translational Lab, co-directed by Jamie Henderson and Krishna Shenoy, for his co-first author paper published in Cell. This study used microelectrode recording arrays to revisit motor somatotopy in 2 subjects with tetraplegia as they made or tried to make various movements of the face, leg, and arms. What they found, surprisingly, was that the so-called hand knob area of the premotor cortex in people with tetraplegia is tuned not just to one region of the body, but to the entire body. And this contradicts traditional views of human motor cortical organization. They describe what seems to be a compositional code that links all 4 limbs together, potentially facilitating skill transfer. This is especially exciting because with whole body tuning, it then becomes possible for a discrete brain-computer interface to accurately decode all 4 limbs. This work and paper are likely to help the field entirely rethink how the motor cortex is organized overall. First place this year goes to Sayaka Inu from the Shah Lab for her paper in Cell. Behaviour, as we know, is inextricably linked to internal states, but little is understood of the mechanisms that govern this link. In this beautiful study, Sayaka showed in mice that estrogen acts on a genetically specified set of hypothalamic neurons to rewire the connectivity dynamically across the ovulatory cycle. She showed that the rise of estrogen that controls ovulation also induces a threefold structural increase in the connectivity of a specific projection to its target, that this increase is electrophysiologically meaningful, and that inhibiting these connections does away completely with female sexual behavior. Her discovery of large-scale dynamic rewiring of connections in the adult brain is paradigm shifting, as it raises the exciting possibility that the adult brain may rewire equally dramatically in other physiologically relevant states. So at this point, usually in a normal year, we would have everybody up on the next to the podium so that they could look awkward and everyone could look at them and check them out. But it's not an ordinary year, so they get away with having just their mugshots here. And I'd just like to congratulate everybody again for their fabulous efforts. It's always very exciting to read the papers, and we will be watching you all going forward in your very exciting careers. So congratulations again.